My name is Damiano Barone. I'm a neurosurgeon and a scientist specializing in functional and peripheral nerve surgery. I'm an assistant professor at Houston Methodist and Weill Cornell. Today we're going to focus on a topical argument, which is brain-computer interfaces and neural interfaces, which attracts quite a lot of media attention, but is often not very well explained. So today we're going to go in the details of what brain-computer interface is and some of the other terms that people use to describe it. And then we're going to try to work out how a brain-computer interface works, what is able to provide to the patient today, what we'll be able to provide to the patient in the future, what are the challenges on translating this technology into the clinic. So let's start with nomenclature. There are so many terms that get used around the term BCI, brain-computer interface. They all mean something slightly different, but they also get used interchangeably. So the main umbrella term is neurotechnology. Neurotechnology represents any type of technology which is used with the nervous system including brain, spinal cord, peripheral nerve, muscle, but it will include technology like surgical robots or wearable devices or imaging tool like an MRI or CT to imaging the brain. That's all neurotechnology. One branch of neurotechnology is neural interface. So what is a neural interface? Neural interface is the interaction between the human brain and electrodes. So the electrodes can stimulate the brain, so sending electricity to the brain, or can record from the brain. So there is stimulation and there is recording. This interaction, this electronic conversation between the brain and the electrodes is what we call neural interface. As part of neural interface, you have BCI, which is brain-computer interface. Brain-computer interface tends to be more of a one-way system. Is the recording side. Its use is that taking information, electronic information from the brain to a computer and to then control something. For example, a cursor on the screen or a speech software. A similar term to brain-computer interface is BMI, brain-machine interface. Again, they're used quite interchangeably. But the real meaning of brain-machine interface that's been given is the recording taken from the brain is used to control, for example, a robotic arm. So controlling something like an exoskeleton, something that is outside the computer, a machine. Then there are all the terms that we need to familiarize, which is bidirectional. So if the brain-computer interface is able to do both stimulation and recording, that's called a bidirectional neural interface. Now, if this recording inform the computer to do something. So we take information from the brain, this message is analyzed, and a feedback is given automatically to the brain. That's what we call it closed loop. That would be a closed loop BCI. So these are all terms that we'll be using, and are all quite important in understanding how neural interface work. But let's go a bit more in the details. So when we talk about neural interfaces, there are not one single type. There are several types, and let's go through the different typology. So the devices can first be categorized in non-invasive and invasive. Non-invasive devices, for example, people are very familiar with EEG. EEG are basically electrodes placed on the scalp of the patient. So those are the least invasive, but also the least precise. Then when we go in, we can have other two types of device. We can have a device that is installed just on the skull of the patient, or a device that is on the surface of the patient. So the surface device is called ECoG, electrocorticography. It's still not penetrating the brain, so it still is invasive, but minimally invasive, just because it doesn't go in the brain parenchyma. And then when we start going in the brain parenchyma, we got two main devices. We got the intracortical device, which basically has small spikes that go inside the first few millimeters of the cortex. And then we got the deep BCI, which go centimeters down to the cortex. They all have a function and they all have a value. More superficial you are, more you sample from a large population of cells. So you sample from many neurons. More close you are to the parenchyma, in particular if you're inside the parenchyma, you can go and sample from one single neuron. Now, the advantage is when you go in one single neuron, then you get straight to the information that neuron does. When you start taking information from a population of neurons, then you have to start guessing and interpreting what that single neuron in that population could do. And for certain function, you need one. For others, you need the other. Similar, in the spinal cord, we got two main devices. We got an epidural, which sits on the surface of the spinal cord. This is commonly used in clinical practice for a spinal cord stimulator. Or there are intraparenchymal devices, similar to the intracortical device, but they're really only used in the research world. In the peripheral nerve, we've got many different types of devices. The comparison to an ECoG would be a cuff device, so a device that goes around the nerve. Then we will have an intraparenchymal, so intranerve device that will go in. And then we got even a more invasive type, which is called the regenerative device, where basically the is inserted after cutting a nerve. The nerve is cut, and the device is put in the middle, and the nerve will regrow through the device. Again, it's the most invasive, but also the one allowing the best recording inside the nerve. So these are the different types of devices. But let's see how specifically they work. So we have a neuron. So this is the cell body, this is the dendrite, that's the axon. We know that the neuron is an electrically active cell. So being an electrically active cell, it causes perturbation around its membrane, and this perturbation is electrically active. It's ions going in and out, causing a differential in potential. So if we put an electrode near the cell, we will be measuring what is this differential in potential. What will look to us when we do the recording is just squiggly lines. These squiggly lines needs to go and be amplified. They are very small squiggly lines, so they go something called through an amplifier. And then after the amplifier, they get digitized to then become something that is intelligible and usable for us to make an interpretation of the recording. Now, if you have one cell, then you have a single cell recording. But if you have a group of cells, then what we actually measuring is something called LFP. We're measuring a local field potential, which is summation of all these differential from a large group of cells that's, again, squiggly lines, they get amplified, and they get digitized, so we can use it. So once we record the local field potential of the single cell, then those get digitized in the computer and they get implemented in an algorithm. We know that this signal comes from a specific part, for example, of the brain, and we know, for example, if they're coming from the speech area or the motor area. What we do is also recording the patient talking or the patient moving, and we try to associate these two functions together, so to interpret what this local field potential signature means. For stimulation, we do the opposite. We can control different aspects of how the electricity is delivered. We can control how much electricity we deliver, for how long we deliver, and how frequent we're going to deliver this signal. So we can control amplitude, duration, and frequency. And that is really the language of the electrical signal. Every different aspect modifying any of these factors is a different letter of the alphabets. And we have to know how to use that to then talk to the brain. And that's the language of the brain. That's how we talk to the brain, by using just these three factors and altering over time. We can deliver these to a group of cells, or again, we can deliver to a single cell, more commonly to a group of cells. Now, with this stimulation, we can do two things. We can activate this group of cells, but also we can stop this group of cells. So we know, for example, by stopping certain part of the brain or the nervous system in general, we can affect function. For example, that's how spinal cord stimulation works, by blocking pain signal, or deep brain stimulation by blocking certain pathway to avoid, for example, the patient having tremor. So what type of neural interface we have in the clinic today? Well, we have few that are actually quite common, people know, certain others that are a bit more niche. So the one that probably many people know are the DBS, the deep brain stimulation, and SCS, spinal cord stimulation. Strictly speaking, they are not brain-computer interface, they are not BCI, but they are a neural interface. This is still a conversation between an electrode and the nervous tissue. When we go in the world of BCI, we move towards this type of electrodes, so either the intracortical electrodes or the ECoG electrodes. Those are the ones meant to record at high density, high fidelity, and those are the ones that have been used for example, speech interpretation or motor. So they are the speech BCI and motor BCI. Again, these are still in the research realm, but applied to patients. There have been now many examples and many groups that have demonstrated how to use both this technology, for example, for speech or for motor intents, moving an electronic limb. On the spinal cord, we have neural interfaces that are being used not just for pain control. Spinal cord stimulation nowadays also be used for motor rehabilitation, helping people with paralysis. And the peripheral nerve wards at the moment are still being used mostly for pain neuromodulation, but there are many examples in the research side, in particular in animal models, where we'll be using PNS devices, nerve devices, for motor restoration, sensory restoration, autonomic control. For example, controlling bladder bowel function in patients with spinal cord injury. This is where we are now, but where are we aiming to go? Well, at the moment, we can use these devices really to synthesize speech, possibly control a robotic hand, moving a cursor, and this still are life-changing for patients. If we can achieve this function, even just those dysfunction, those will be really life-altering or life-changing. But we want to do more. We want to restore function completely. We want patients to go back to their previous state, to revert to what happened after a traumatic brain injury or a spinal cord injury. But to achieve that, there are challenges. And let me tell you about a few of these challenges. So there is a biological challenge. So let's think this is part of the brain. This is an electrode. Once we implant something in the body will have a reaction. The reaction to an electrode is, we call it, or any implant in the body is called FBR, foreign body reaction. The way that foreign body reaction works is by the body will start secreting collagen. Well, we start first activating the inflammation pathway. Inflammation pathway will push the fibroblast to produce collagen, which will start producing scarring. Now, the scarring will get bigger and bigger, and the previous contact between the electrode and brain, which allowed us to record from the neurons, becomes noisier and noisier until basically we cannot record anymore from those neurons in the brain. So until we can find a solution for scarring, then long-term BCI becomes a problem. Many of these devices have been used, for example, either for very short term during surgery or using manipulation on new devices. But so far we struggle to have a device that can stay inside the body for the lifetime of the patient. And that's what we want. We want a neural interface that does what I said, but it does it for the lifetime of the patient. It won't be good enough to have a device that only stays for 1 or 2 weeks. That will not be something that will be clinically acceptable. That's the biological problem. Then we go engineering problem. These devices at the interface are small, but on the back end of these devices, there are massive computers required to run it, because the algorithms are really heavy. Again, we cannot have a patient carrying a massive computer on the back. We need this to be miniaturized so they can be implanted together with the device. The algorithm can be scaled down. That has been something that we'll be doing, but then there is a second engineering problem, which is energy. This is very power consuming. They require quite lots of processing, so the battery needs to be there and available. Nowadays we have rechargeable batteries. Battery can be recharged while inside the patient. By this point in time, they require so much energy that the battery will need to be recharged all the time. So it's something that is a problem. Now, certain groups are working on systems that sit outside the body and are connecting to the body wirelessly. So for example, the battery and the processing is outside, for example, wearing a hat or wearing a belt, so you have a bit more space, but again, it's not ideal for the patient. So these are still challenges that need to be overcome. And then let's not forget about safety, privacy. This is a window into the brain, as people will say. Now, it's not like any of these devices at this point in time can read thoughts or examine memory. It's still very specific to that function, but are still connected to a computer system. So potentially there are risks of safety, biosafety, and these need to be explored together with ethics. The long-term goal remains very simple and very human, is to give people the chance to communicate again, move again, speak again, to really go back in their life. We're looking for restoring function rather than enhancing function. And Houston Methodist is investing quite a lot of effort in developing these technologies in my lab, in other physician and scientist lab, and I will be talking more specific of every single technology in future videos. Thank you.